Hi, I'm Dr. Lalita Calabria. I'm a faculty member at the Evergreen State College in Olympia, Washington, and I'm a lichenologist who studies biodiversity and conservation of North American lichens. And today I'd like to share a bit about my research and how I contributed to the Science Stories project with book artist Anne Greenwood-Rioseco and Daniela delMar. I'd like to begin by defining what a lichen is. Here's a cross section of a scanning electron microscope image of a lichen. And what you can see is that a lichen is composed of a fungus and an algae, a fungus and a cyanobacteria, or both. The majority of the body of the lichen, which you see here, is composed of the fungal partner, which provides a habitat for the algae to live in and helps to regulate moisture and provide protection against UV light, whereas the photobiont partner, which is the algae or the cyanobacteria, is able to provide food for the fungus through photosynthesis. Recently, there was a discovery made that lichens are not just composed of the fungal partner and the photosynthetic partner, but also a third fungal partner called the basidiomycin yeasts. And this was an amazing discovery that helped scientists reframe their understanding of lichens and stop thinking of them in this reductionist view as lichens are a fungus, one algae, and what those individual parts contribute, but returning to this view of lichens as a whole, as an ecosystem and the microbiome that occurs that we can study to better understand their role in ecosystems. So one of the aspects that I contributed to the collaboration for the Enmeshed project was looking at lichen biodiversity and conservation and thinking about rare lichens. Here's an example of a rare lichen that has a fascinating life history. This lichen has a very restricted distribution in Oregon and Washington with only just a few sites where it occurs. The variation coccinea is a much more rare variety, and no one knows why it's red. Why does it produce these bright red pigments, and what adaptation do these pigments confer that make this phenotype continue to exist in these populations? So right now, this lichen, this red one, is considered critically endangered in Washington with a very limited distribution with a questionable taxonomy. And so my research group set out to find out what pigments were being produced and why, and which member of the lichen symbiosis was actually producing these pigments. And our aim was to better understand the distribution of this lichen in order to inform conservation and management practices. So this is a map that shows the distribution of the rare variety of the lipstick lichen, Umbilicaria fea variation coccinea. And you can see there's only 2 known locations in Washington and several sites near the Siskiyou National Monument in Southern Oregon and Northern California. So the first step was to collect samples from the field. And this is my student, Mac Patton, who you can see is collecting these samples down in the Siskiyou National Monument. You can see here with my other students, Jesse Grave and Eli Livesey, who were involved in the purification of the red pigment, which you can see here. The next step was to analyze the metagenome. So I talked about the microbiome of lichens, and this is something that allows us to not only study one of the organisms in the symbiosis, but to get an understanding, if you took a sample of this lichen, what are all the genomes, are all the species present in that symbiotic organism? And with this technology, we can better understand the metabolic pathways that are able to produce these pigments. What and why are these pigments produced? So this is some graphics that were created by Gwendolyn Root using a software called KBase. And this is just one part of the microbiome, focusing on the bacterial samples along with some of the eukaryotic samples in the genome. So this is one way to visualize the data. And at various different taxonomic levels, we can start to get an understanding of what this symbiotic organism is composed of. Umbilicaria fea var coccinea is not the only rare lichen that we have been working on in the Enmeshed Science Stories project. Here are some of the other lichens that have been featured in this project. We have Sticta arctica, Bryoria bicolor, Usnea rubicunda, Xanthoria elegans, and Loberia oregana. And it was wonderful to collaborate with Anne, who was able to participate in a lichen dye workshop with my students, where she actually took the lichen, Latheria vulpina, the wolf lichen, and was able to dye some of the textiles used in the book for the Enmeshed project. So it was really fun to collaborate in this way. And we're super excited to share more about our science stories with you and for you to learn more about lichen conservation and biodiversity. And I hope you enjoy these beautiful illustrations by Leanne Smith Lucero of these rare lichens. Thank you so much.